And can anybody please tell me the thing like till what content we are done yesterday? Which database we have started and what part we have done yesterday? Hello? Am I audible properly?
Yes, Priyanka, you're audible.
Okay, sir. Okay, so I'm sharing my screen now. Okay, so yesterday we just have gone through the UCSC questions and we were remaining with the two exercise questions. And these questions were retrieve the whole genome sequence for the mouse, this one, and retrieve the complete annotation of the chromosome 1 of the mouse. These two questions are left. Another questions are done for you, all of you. So if any doubts are there in this exercise questions, you can ask me. Otherwise, we will move to the next question then. Any doubts for the rest of the questions? Hello. Okay, fine. So let's go for this question. So we have to retrieve the whole genome sequence of the mouse. Okay. So can you please tell me again the retrieval is there means where should I supposed to go now? Sorry?
The browser at UCC, we were doing the human browser.
No, not a genome browser because here we have to retrieve the data means we have to go to the table browser. Yes. Just give me a minute. Like some people are having some issues in joining the session. Okay. So here we have to retrieve means we will simply go to the table browser and we have to retrieve a complete genome sequence of mouse. So in the UCSC database, I have told you from the download section how you can download the complete genome. That might be clear to you all. I think somebody's mic if mic is on. Can you please off it? Ajisha ma'am? Yes. Okay, so we will simply open the UCSC database now. And let's open the table browser of it. So tools, table browser. Now here again, clade will be mammal, genome will be mouse, assembly will be current assembly. And whenever you are downloading the sequence from the table browser, so you have to choose gene and mapping and sequencing, because it is related to the complete genome sequence only, right? So mapping and sequencing, you have to choose the track as assembly. If you want to download the complete genome of the mouse, select here genome. Output format will be sequence. And then if you want to download it, try to provide here the file name and say get output. Your file will be downloaded. I think this question is done. Repeat regions are also done. Okay, this question is left. Nobody has told me that this question is done.
Yeah,
This was
actually,
done.
yesterday you marked it, but why this is
No, I haven't saved it actually. So this question was left actually.
one more is there, one more is I think the
Which
last
one?
two were left.
Yes, this one and complete annotation. These two are okay. So let's, okay, again, not an issue. We just have revised this question, not an issue. Let's move to the next one. So here we have to retrieve the faster sequence of the first 10 transcripts. So if you remember this transcript sequences, you can retrieve it from the NCBI also, isn't it? But now in the NCBI, what you need to do, you have to retrieve a transcript for a transcript sequence for first transcript, then second, then third. Likewise, you have to choose it separately, isn't it? So now here in the UCSC, within 1 minute only, you will be able to retrieve the first 10 transcript sequences in one go. And how you can retrieve it? With the help of bulk data retrieval options. Okay, so whenever you are going for a bulk data retrieval, you have to go for a bed format. Now, what is this bed format? So let me try to open this mouse genes in Excel sheet. Like whatever the file we have downloaded yesterday, because in that file only we were having the transcript and gene information, isn't it? So open this file from your side also. It should be delimited, tab separated, next and finish. So I'm having this file with me. So what you can do is, here you can see we are having the all the details related to the transcript over here, right? So whenever you are going for the bulk data retrieval, as I have told you, we have to make the bed format, isn't it? How you can make the bed format? So bed format will be generated. If you remember the position format also, there also we are mentioning the chromosomal location, start coordinate and the end coordinate, right? So for the first 10 transcript, I'm just going to copy this chromosomal location, transcript start and end coordinates. Strand information is not required, but I'll just delete it in the next step. I'll copy them from here and I will paste it in the new sheet, okay? And this strand information I will delete it because that is not necessary in the bed format. Okay, so here you can see I'm having the chromosomal location. Then we are having the start coordinate and end coordinate. Is that clear? So maybe you can just copy this headers also, but in the bed format, it's not necessary to mention your headers also because it's just focus on this particular part. Okay, so whatever the thing we have made it here, this is nothing but our bed format. Is that clear? Everybody, is this point is clear to you all? Okay. So now, if you want to retrieve the sequences, maybe for the 80 transcripts, or maybe you want to retrieve it for the exons. Just the thing is that you will require the chromosomal location, start and end coordinates, paste it in the new sheet, make the bed format and copy it. Okay, I have copied this bed format for the 10 transcripts. I will go back to the UCSC, and now whenever you are making the bed format, whenever you are going for the bulk data retrieval, directly go to the my data and custom track. Okay, because this custom track type of search is used for the bulk data retrieval. So my data, custom track, and here in this particular page of clade genome, you can select it. Assembly also you can select it. And then here in this search box, I'm going to paste my bed format. Okay, first train transcript bed format, and then I will submit it. And yes, again, if you want to visualize this transcript, you can view it in the genome browser. If you want to retrieve the data, you can just go for a table browser. Okay, so as of now, we also want to retrieve the sequences. So I will view it in a table browser and say go to first annotation. Till here, the things are clear. So whenever further you are submitting the bed format in a My Data custom track, do not change anything in the select data set. It will automatically update. Okay, and as if you will select here the position, so only the first position of your bed format will be selected, only the first sequence will be displayed. So whenever you are providing the bed format, try to select here genome. So all the positions which you have provided in a custom track, or like that bed format, that all will be taken in the genome. Okay, output format again you can choose according to your need, but in the question we have to retrieve the sequence. So I will choose it as a sequence. I don't want to download the sequences, I'll just display them. So get output and get sequence. You can see here I'm having the first transcript sequence, then second transcript sequence. Below, if I will just scroll it down, I'll be having a 10 transcript sequences over here. Okay, the sequence is too very large. But I hope this is clear to you all. This is how you can download different sequences in one go by just providing the locations in a bed format. Is that clear?
Yes, ma'am.
Okay. So, one more thing I want to mention it out over here, that check it out these things now, whether we have retrieved the correct information or not. Like the cross checking also needs to be done, isn't it? So if you're like, if you might know about the bulk data retrieval from the UCSC, but if you are not knowing this thing, then you are not retrieving the correct information. Okay, can you spot it here? The bed format we have provided for these locations, right? But the first position, if you will see now, end coordinate is matching, like 3172348. It's matching. But the first coordinate, can you spot it? It should be 3172238, but it is starting from the 39th position here, right? Here also, if you will see the second transcript, it should be from the 9955, but it is starting from the 56. End coordinate is fine, but the start coordinate is increasing by 1, isn't it? So we are losing the first nucleotide base from here, right? Because why this is happening? Because the UCSC is having the tendency to increase the one coordinate at its start position. So that's why there is one thing, like whenever you are making the bed format, try to minus the coordinate here only. Okay, whenever you want the sequence from this position, minus it 1 from here only, so that from the 38th position, you will be having the sequence over here. That is what the trick you can apply it. How you can minus 1? Say is equal to, select the start coordinate, minus 1, enter it. Then you can autofill the formula, remove the formula, cut it from here, paste it here. That's it. Now, if you will submit this bed format by minusing the 1, then you will be having the proper sequences. You will not lose your first base. Okay. So yes, this thing I wanted to mention here is that whenever you are going for the bed format, try to first of all minus 1 coordinate from the start coordinate and then paste the bed format so that you will not miss the first base also. Is that clear? Do you want me to show by submitting this also?
Yes, ma'am.
Okay. So whatever the bed format we have made by minusing 1, I'll copy it from here. I'll go back once to my UCSC, my data, custom tracks. We'll add the custom track over here. Submit it. And then we will view it in a table browser. So go to first annotation. Whenever you are submitting any custom track, you don't have to change anything in the select data set. Here it should be genome. Output format will be sequence and say get sequence, get sequence. Now you can see if I'm minusing 1 coordinate from the start position, so I'm getting my sequence from a proper locations now. Right? Yes. So that's what about this question. If any doubts are there in this bulk data retrieval, you can ask me. Hello? Okay. Let's move to the next question now. So the next question we are having as retrieve complete annotation of chromosome 1 of mouse. Okay. Annotation is nothing but the extra information. So actually we are having a 2 standard annotation file formats, and these are what GTF file format and GFF file format. Okay, GTF is developed and maintained by the UCSC, and GFF file format is developed and maintained by the NCBI. Content in both of the files will be having the same format, but just the thing is that they have they are being maintained by the different databases. Okay, so here as we are dealing with the UCSC database, so the annotation is representing what we have to go for a GTF file of chromosome 1 of mouse. Now again, whenever you are downloading these genome files or the GTF files, obviously they are going to have a larger sizes. So do not directly download it in your systems. Okay. And do not open them also because it may slow down your system. Now, in case if you're having a proper configuration in your system and you wish to download it for further applications, so I will just give you an overview how you can download it. Okay. So as retrieve is there means again, obviously we will go back to the table browser, tools, table browser. Again, clade will be mammal for our mouse, for our question. Genome will be mouse, assembly will be this one. And whenever you are going for a GTF file, so GTF file is what extra information and it will be not coming in terms of sequences. So that's why we have to go for a gene and gene prediction, and track you have to choose it as a NCBI RefSeq. Then here they are asking us to get the GTF file for the chromosome 1. So you can select it here, the position as a CHR1. And then here the output format, you can select it as a GTF, which is gene transfer format. If you want to download it, provide the file name with .gtf extension. Will not download it, I will say get output. So just see this file. I'll explain you the content by referring the another link. I know it is going to stuck in between, so that's why I'll not explain you here. But just see, this is how the GTF file will looks like. I'll go back because it is going to stuck now. So I'll just explain you this GTF file content with the help of one another web links. GTF file format. I'll open this in symbol link. And you can see the GTF or GFF file. Both of these are the standard annotation file formats. And these are going to have a same kind of content. And they'll just mention the fields. Fields are nothing but the columns. Okay, fields must be tab separated. So now it will be having the 9 columns actually. So first you are going to have the sequence name, means whatever the IDs you are having it, or the chromosomal location. Then source means from whichever the database we are downloading the information, or they are taking that information, that source name you are going to get it there. So here, see, it got stuck in between, it cannot open. So source, if you are downloading it from the NCBI Ref6, so it will give you the source as a NCBI. Feature is what? As this annotation file format is nothing but the extra information. So how you are going to get the extra information? In terms of feature. So in the third column, you are going to have the extra information. Maybe it can be gene, it can be exon, it can be intron, any kind of extra information will be present in the third column. Then in the fourth column, you are going to have the start coordinate of your feature. Then in the fifth, end coordinate of your feature. Then you're going to have the floating point score. Then in the seventh column, strand information from like whether this feature is lying on the forward or reverse strand. Then you're going to have the frame information in the eighth column. And in the ninth column, you're going to have the attribute, means what? Will be having the gene ID, or you will be having the transcript IDs over there. Okay, now if you will open that GTF file again, you will be able to see the same attributes, same fields over there. Okay, but as the information is too way larger, so it will stuck in between. So that's why I'll be not opening it. Okay, is that clear? Yes, okay. So, yes, that's it all about the UCSC questions. If anybody is having any kind of doubts in these questions, you can ask me now, because further we are going to start with the next database then. Okay, so as nobody is asking the doubts, I think all the questions are clear. Or might be after the session, you can practice it out the things. And if any doubts will be there, you can ask me in the next session also. Okay, so yeah, that's it all about the UCSC database. So what we have studied till now is we have studied about the gene database from the NCBI. There what we have did, we just have took one gene and we have tried to retrieve the possible information related to that one gene, right? In the UCSC, what we have done, we just have retrieved the bulk amount of data. We have seen how we can visualize the data in the UCSC, isn't it? That is what the main purpose of it. Now we are going for the next database, which is used for the literature search. Let me check this thing out first of all. We'll be dealing with a Pubmed database. Okay, now we are going to start with the Pubmed database. Do you have any idea related to the Pubmed database? Anybody? Anybody is having any idea related to the Pubmed database? Why all of you are not responding? Are you not getting the things? If you are not understanding the things, please let me know. I can revise the things for you if you are not understanding. Yes, it's a database for the biomedical literature. Because whenever you're working on some topics, or whenever you're working on some, whenever you're performing the research work, so first and foremost thing is that you should know all the things related to your topic, isn't it? Like if you're working on some disease, so you should know till today what research has been done on that particular disease, isn't it? And from where you will be able to know that? From the literature search only, means from the research papers. And that research papers you can directly find it in the PubMed database. So might be most of you have retrieved the research paper from the PubMed database, but only one person has responded me for this PubMed database. Okay, not an issue. Let's go for the introductory part of this PubMed database now. Okay, so yeah, most of the people will be aware about the PubMed database. I also thought that only. So let's go for the introductory part and then we will go for the exercise questions of it. Okay, so the PubMed is a free resource which is supporting the search and retrieval of the biomedical and the life science literature as correctly told by the user. Then the PubMed database contains more than the 36 million citations and the abstracts of the biomedical literature. So basically this PubMed database will be having the cited data also, and you will be having certain research papers which are freely accessible. You don't have to ask for the permissions for it. But yes, these are provided to you in a, like if you are not aware about it, I'll be telling you how to go for that filters and how you can retrieve the such kind of freely accessible data. Okay, then here we just have provided you the link for the PubMed database. Either you can use this link, otherwise simply in the browser also you can just type it as directly PubMed database and it will open that particular database. This is how actually the PubMed database looks like. Home page will looks like. We are going to deal with that. But first of all, let's see, because your different databases are going to store the information in a different format. Okay, so here the PubMed database, it does not include the full text journal articles. Okay, it will be having the abstracts for that. Maybe even if you are having the free full text journal articles, but you have to ask for the permissions like this. Then PubMed primarily contains the information about the biomedicine and the health fields and related disciplines such as life science, behavioral sciences, chemical sciences, and the bioengineering. Like most of the life sciences fields are getting covered here. Then most important part over here is PubMed facilitates searching across the several NLM literature resources. So basically this NLM is developing is controlling our PubMed database. Okay, our PubMed database is developed and maintained by the NCBI and this NLM only. So from here they are taking the data and how they are storing it. So basically in the PubMed database, the information or the data will be stored in a midline, PubMed Central, and the bookshelf. And again in the midline, which kind of information is present? So here you can see midline is the largest component of the PubMed and consists primarily of citations from journals selected for a midline. and the articles indexed with a mesh. So mesh is what? It is one database related to the medical field, which will be having the medical subject headings, and the data will be totally curated. So in the Medline, most of the time you will be having the cited data only. You cannot read that data freely. If you want to read it, so you have to ask for the permission to the author. Okay, so such kind of curated data you can get it here in the Medline, for which maybe you have to pay for that information. Then in the PubMed, you will be able to get the freely accessible data also, like all kind of data will be stored in the PubMed Central, even though the data will be curated or maybe cited data or the free full articles also you will be able to get it in the PubMed Central. So that's why for the exercise questions also we will be using this PubMed Central only. Then we are having the bookshelf for your interested topic. If you want to get the books, so that books citations, again uh, you have to again buy that books from this bookshelf. You are having that separate option where you can find the books related to your topics. You can just cite it that books likewise. Then, yes, here again the same thing which I have told you here. The Medline is the main part of the PubMed, which is an online searchable database of research literature in the biomedical and the life sciences. Medline is authoritative in the sense whatever the data you are having in the Medline, that you have to ask. If you want to read it, you have to ask to the author, like you have to take the permissions of them, maybe you have to pay for it. Then searching, obviously the searching in the Medline also, it's a free of charge. It will be not requiring any registration, but if you want to open that data, then you have to ask the permission. Then PubMed Central is a free full text archive of biomedical and the life science journal literature where you will find all the data. Then Bookshelf is a full text archive of books, reports, databases, and another documents which are related to the biomedical, health, and life sciences. So, likewise, Bookshelf is also present in the PubMed. So, further, actually, we are having the exercise questions. So, that's why what we can do is we'll get familiar with the PubMed database homepage and then we will solve the exercise questions. So let's try to open this PubMed database now. It's actually not that much difficult database. It's easy one. Till then, I'm just sharing you the attendance link. Try to fill it. Okay. I'm sharing the attendance link with you all. Try to fill the attendance sheet also. And then open the PubMed database. This is what the link we are having for it. I'm pasting the link for the PubMed database also in the chat box. Have you filled the attendance? Shall I go for the introduction of this? Okay. So this is how actually the PubMed database looks like. I have told you it's developed and maintained by the NCBI. So you can see it here. It is developed and maintained by the NCBI and NLM, which is National Library of Medicine. Here you are having the logo for the PubMed. Here in the search bar, you can just provide your topic for which you want to get the research articles. And here again, you are having the learn, find, download, and explore options. Learn in the sense, if you want to learn anything from the PubMed, how to retrieve the data and all those parts, you can learn it from here. They will be giving you certain documents for that. Find, if you want to search anything in the PubMed database, then you can use the find option. If you want to download the research articles, that you can download it from here. If you want to explore it in the mesh database that I have told you, mesh is one medical database where the medical subject headings will be provided. So if you want to explore your topic in the mesh database, you can explore it. Okay, so yeah, this is what the basic homepage we are having for the PubMed database. It's not that much complex, but main important part in this PubMed database is that how you can apply the filters. Okay, that is the most important part. So like whoever has like most of you might have used this PubMed database, but you might be not aware about some filters. So that might be new to you. So try to understand those things. So let's go for the exercise question. And the first exercise question we are having is what? How effective is this aspirin in preventing the heart attacks in the elderly people? So here I'll just highlight those things which we can mark it in this PubMed database. So here we are having the aspirin. This is what my search term actually, isn't it? Then we have to search the aspirin articles which are only useful in preventing the heart attacks, right? So heart attack again can be my filter over here. And one more, we have to go for only elderly people, means the people who are having the age above the 65 plus. So for them, we have to get the research articles, isn't it? So now here we can provide one search term as an aspirin and the next we can apply the filters over there, right? So let's go for it. I'll open my PubMed database and here in the search bar, I'll provide my aspirin as an search term. Okay, let's search for it now. Okay, can you see here? For the aspirin, I'm having the 75 results over here. Now let me try to explain you how actually these results are coming and what parameters it is having. So we are having 75 results for this aspirin. Then here you can see if you want to sort it out this data according to the dates or according to its maybe author names or maybe yeah, according to your journal names. So you can just sort it out this data according to this parameters from here. Okay. Then here display say options are also provided. So this is what you are having a normal summary format, isn't it? You are having the header of your particular research article, who is the author. Then you are having the PubMed ID. PubMed ID means what? This PM ID. So we have seen in the gene database and in the UCSC also, for each and every gene, for each and every protein, we are having some unique IDs, right? So here also in the PubMed database, we are having the unique ID for each and every article, right? So this particular article is having this unique ID. If you will search for this particular ID only, they will directly open this article likewise. Then they'll just give you whether this article is freely available or we have to go for a citation of it. So that information they'll mention it. That is what we are having the display as an summary format. Okay, so here if you want to change this display option, you can click on this display option. And if you want to display the abstracts, if you want to display these articles according to the PubMed or PMID, so that options also you can choose it. But most of the time, the summary is fine for us. Okay, so we are having these many results in the first page. We are having maybe 20 results likewise. Okay, this is what the first page out of these many pages. Now, if you will see in this particular left hand side, you are having the filter panel. Okay, so here you are having the year filters actually. So you can just scroll this particular circles from here and then you can be specific about your years. Maybe You just want to get the research articles from um 2020 maybe. So you can just scroll this circle till 2020 and you'll be only able to get the research articles which are published during that years. Okay. Then here you are having the another filters also for the article types and then publication date also. Maybe you want to get the most recent articles which are published in the last 1 year or maybe last 5 years. That filters also you can apply it from here. Species filters are also there. Maybe you're able to see some filters in my system, like in my Pubmed database, but these are not visible in your site. So you can see here you are having the additional filters. Click on that. And again, here also you are having a lot of filters. So try to check these things out. So you just have to select that filter, click on the show, so it will be just shown into your filter panel. You can select it from here and you can apply those filters. Okay, so as of now we just have search for the aspirin, but we just want to get the aspirin for what? Heart attack, isn't it? So if you will search it in the filter panel, you are not having any heart attack option, isn't it? Or heart attack filter. So what you can do is here just provide one operator. We can provide one operator as an and then provide your search term as an heart attack. Okay, then search for it, and now you can see you're just having the 9886 results. Now we just have got the articles where the aspirin and the heart attack is getting matched. Okay, so can you please tell me what is the next filter which we need to apply? Elderly people, right? It's related to the age, isn't it? So age filter we are having here in this filter panel. If you're not able to see it here, that's totally fine. Just go to additional filter. Here you are having the age filter. And yes, from here, maybe if you want to get the same articles for maybe child or maybe infants, that filters also you can choose it according to your choice. Okay. As of now, we have to go for a elderly people means 65 plus years age. So I have selected the aged filter and say show it. So after clicking on the show, the filter will be not applied. It will be just shown here in the filter panel. So click on that filter and then you can see I'm just having a 3973 results. Now we have got more specific about the results, isn't it? Now you can just go for, maybe you can sort it out this data according to the publication date, and you can just read these articles and you can see how much effective is this aspirin in those peoples then by opening these articles. Is that clear? Hello? Okay. Only two people are listening to me. What about the others? Ayush sir? Okay. Okay. So are you guys okay? Like if we will extend the session for a 10 minutes? Because today also we have started the session bit late. So if you're comfortable, we can, yeah, extend it. Okay. Okay, thank you so much then. We will extend it bit uh, more then. Then we are having the second question as construct a search to find the articles for a breast cancer with a PMID. Means what? Whatever the PubMed ID they are providing to us here, we have to identify. We have to cross check whether this article is written for the breast cancer or not. Is it is related to that or not? So what I can do is I'll simply copy this PubMed ID from here. I'll just paste this. Okay. Before you are going for the next search, try to clear these filters. because this filters will be, otherwise that filters will be automatically applied in the next search also. So now here I'll provide my Pubmed ID. Yes, you can provide your Pubmed ID also as an search term. Okay, search for it. And we know that for each and every article we are having a unique PMID. So that's why we are just having a one article over here. So by reading this header only, you will be able to see here the the information is being provided for the mammary carcinogenesis. Mammary is nothing but related to the breast. Carcinogenesis is nothing but the cancer only. If you will search it on the Google, you will be able to see here it is related to the breast cancer. Okay, so yes, whatever the search we have performed over here, that is for the breast cancer. Yes, that's correct. Okay, is this question is clear? Yes. So let's go for the next question. You can also try it out the next question now. So we have to find out the recent articles for the Parkinson's disease which are published during the last 5 years. We have to apply a limits to our search by filtering the results for the human, and the article type should be limited to clinical trial. A lot of filters have been applied here in this question. Like we have to apply in this question. First search term we are having as in Parkinson's disease. Okay. We have to get the research articles which are published during only last 5 years. So this is also one filter we have to apply. Then we have to search the data only for the human species. So this is the one filter, and the article type should be limited to the clinical trial. So these many filters we have to apply it here. Okay. So what we can do is right now I'll copy this Parkinson's disease search term from here. I'll provide it here in the Pubmed database, this search term now. Before that, check whether you have applied any filters. No, these are not applied. I will provide my Parkinson's disease as an keyword. Then let's go one by one for our filters. We have to go for a data which is published during the last 5 years. So this year filter you can apply like two ways are there. Maybe you can just scroll this circle also, or maybe you can apply the filter from here, publication date for 5 years. Just apply this filter. So now I'm just having the 44,000 results. Here also you can see this is automatically adjusted. Then we have to go for only human filter. Again, this human filter, you will be not able to see it here. So you can just go to the additional filter, go to species, select human, show it, and it will be shown here. So select that species filter now. Okay. Now I'm just having 27,000 results now. Then the next filter we have to apply for the clinical trial. Like whatever the articles we have to filter, that should be only clinical trial data. So how you can again apply it? Scroll bit down, and here I'm having the article type as a clinical trial filter is being mentioned. Click on that, and the final results we are having as an 891 results. Okay, so this is how in the Pubmed you can use different filters to be specific about your results. Okay, because obviously we cannot read the thousands of research papers, isn't it? So try to apply the different filters and then uh like use them according to your convenience. Is that clear? Okay. Okay, so basically these three questions are remaining. These questions are also related to basic filters only. So what you can do is try to note down these questions somewhere I'm pasting it in the search box, message box. Okay, and I'm sending you the attendance link also. So try to fill that attendance sheet. Have I shared it with you? Yes, I have shared it. Okay. So I hope you have filled that attendance also. So try to perform these three questions after the session. And if any doubts will be there, we will be cross-checking it tomorrow. Okay, it will not take more than 5 minutes. Okay, so if any doubts are there in the Pubmed database, you can ask me now. Hi ma'am, could you please kindly upload the yesterday's and today's recorded lecture by tomorrow? Would like to make some revision. Okay, fine. We'll update the sessions. We'll upload it. Not an issue. We'll upload it. I'm stopping the recording.